Hi, welcome to this module. This module will be quite an interesting one. This is an experimental module. In this module, we will be exploring a simulated drug screening scenario where we use a microfabricated electrode to understand how a drug testing, cancer cell drug testing could be done. Professor Hardik would have covered several aspects of cancer screening and cancer diagnosis devices. So, you might have heard about breast cancer, oral cancer and several other types of cancer. So, today what we are going to do is cancer can occur also in the brain. So, it is called glioblastoma, and what we are trying to do is we are trying to see how a drug screening or drug testing of cancer cells could be performed. We will not directly do the entire drug screening scenario, but try to show it to you in a very small module so that you understand the whole process. So, what is the whole process? Before we get into the experiment, you need to know the basic things. So, what happens is that let us say a patient is diagnosed with cancer. They go and if they are almost certain that it's a cancer, they take a biopsy, which is a small section of the suspected cancerous tissue from your brain. We are talking about brain cancer, so they will take a small section of tissues from your brain and they do some tests on it, okay, and some tests will be directly on the tissue and some tests will be on cells derived from this tissue. So, this tissue is called primary tissue because it is the first from the site of the person, and from the primary tissue you can crush the tissue, extract the individual cells from the tissue, because why? Tissues are made up of cells, right? Cells are the individual living blocks, okay. So, you can crush the tissue, extract the cells and then do a culturing of those cells. What is culturing? Culturing is where you extract the cells, you start with let us say 100 number of cells, you add growth environment that is favorable or susceptible for the cells to grow, which is usually called a media, onto these 100 cells, and you allow them to grow and they multiply, and you have a culture. These cultures are usually done on cell culture dishes, and behind me you can see an incubator, that is where cell culturing is done. So, you can see there, that is an incubator and that is where cell culturing is done. The parameters that are used for cell culturing are the same as the parameters that are existing inside our body. Our inside body temperature is 37 degrees Celsius. We have 5% CO2 dissolved in blood and we have a 95% relative humidity. So, this environment is maintained inside the incubator so that cells can grow in almost similar human inside the body conditions. Okay. So, what happens? Patient comes, patient's tissue sample is taken, the tissue is crushed, cells are extracted, media favorable conditions for growth of cells are added and the cells are cultured in a dish. Okay. And then conventional drug screening scenarios, what happens is that some candidate drugs will be there which the doctor wants to give to the patient. Okay. Instead of directly giving to the patient and looking for outcomes, what they do is they add the drugs onto these cultured cells and see over a period of time, let us say 7 days or 14 days or 21 days, and see how the cells are dying or are the cells able to survive and proliferate. What is proliferate? Proliferation is the growth and division of cells. Okay. If these processes are inhibited, and when we measure the extent of inhibition of these processes, then we call that the drug is efficient or the efficacy of the drug is good. Okay. This is the conventional drug screening scenario. What we are trying to do is we try to employ this method on a micro-fabricated platform, micro-engineered platform, and try to reduce the time of testing. Okay. So, what we do is, you might have seen the electrodes that we use. So, we have some drug screening device that we have made, which is an interdigitated electrode structure on which we have made a definite well that can contain a volume. Okay. And then what we do is so the cells, when they grow on the surface of the dish, cell culture dish. So, I will show you cell culture dish. So, this is a cell culture dish, this is a 35 mm dish. Okay. So, cells can be cultured on this, and there is also a T25 flask. So, I will show you the flask. This is T25. So, this is a T25 flask, this is called a cell culture flask. and what you see is actually whatever we are going to use today, the cells of this are there in this. It is the color of this shows the color of the media. Okay, when the media is fresh, that means when the nutrients in the media are not consumed at all, the media will be pink in color. When it slowly gets consumed, the pink color reduces, and when it is totally consumed, when it is depleted of nutrients, it becomes yellow in color. The media that we are using is called DMEM, which is Dubocol's minimum essential medium. It is a concoction prepared by one scientist long back, which has been known to be very good for cells to grow. Okay, so another interesting thing that you need to be aware of is, okay fine, this is a T25 flask, this is a 35 mm dish. Cells can be cultured in both. So let me keep this aside. Okay, so another interesting thing that you need to know is that when the cells are cultured on such normal dishes, cells have the tendency to attach to the surface of the bottom surface of the dish. This is called attached growth. Cells by default grow only like that. Okay, so even if you do shaking, you try to remove the media, cells will still be attached very strongly to the surface. This is because of their cell addition, cell attraction. They have a lot of forces acting between cells to keep them together. These are the building blocks by which how cells come together and form tissues. Okay, but for us to do drug screening, we cannot take such attached cells because you cannot take them out. So there is a process called trypsinization, where we add an enzyme called trypsin onto the cell mat of cell growth, which breaks these cell to cell bonds, and the cells get out of this attachment to the surface of the dish, and they enter into a suspension, but they don't die. So this is a non destructive way of making the cells detach from the surface of the culture dish. But this process is necessary for us to do our drug testing. So before we do the drug testing, we will have a culture dish like this, and then it will have cells attached to the surface. Okay, then it will have cells attached to the surface, then we will add trypsin. Okay, then that will cause the cells to enter into a suspension. This suspension of cells we will transfer into an Eppendorf tube. I will show you what is an Eppendorf tube is. So this is Eppendorf tube. Okay, this is where you can easily transfer cells, drugs and all. This is a 1.5 milliliter Eppendorf tube. We will transfer it into an Eppendorf tube. Then using micropipettes and tips, we will load these cells onto the microfabricated wells which are used for drug testing, and then we will see how drug testing can be performed. Okay, so this is one part. Now how are we going to do this whole experiment? So I am going to divide this into three parts. One is, first we will see what cells are we trying to work on, which is glioblastoma cells. So first we will see attached cells, how they are, directly by looking at the T25 flask, and we will see the bottom surface of the cells and see how the cells are looking. Then we will off the video, we will do trypsinization and show you after trypsinizing what will be the liquid that looks like. We will transfer it into Eppendorf tube. Okay, and then use those to finally test using impedance based drug screening of the cells. Okay, so let's get into the first part. We will see cells attached to the bottom surface of the T25 flask. So let's get to the first part. I have kept the T25 flask, which has the attached cells, on an inverted microscope and kept it focused on the cells so that you can see it. First let's look at the T25 flask. So you can see the T25 flask kept on the microscope. This is an inverted microscope that we have. Okay, if you see the flask is there, you can see the cap of the flask, right? So we have seen that it's 5% CO2 is maintained inside the incubator, no? But then if the flask is closed, how does the oxygen supply, the CO2 supply get circulated? So if you see, there are openings here at the tip of the cap, so there is a filter there. So this filter is used for gaseous exchange. If gaseous exchange does not happen, the cells will die. So this is a very custom made flask, especially made for cell culture purposes. Okay, so that's just a detour. So now let's come back. So we have kept the T25 flask. Cells are attached at the bottom surface of the flask. I have focused it. There are lenses below. Now it is kept in 20x objective lengths, 20x magnification. So I can see it through the eyepiece, but for you, I have focused it onto the computer screen. So let's look at the screen. So you can see the screen, you can see lot of cells here, they are elongated very nicely. You can see the elongated morphology. They are elongated because they are attached to the surface of the flask, and they are trying to spread themselves out and be as flat as possible. Like physics, they are trying to be at the level of minimum entropy, okay, and they are spreading themselves out. That's why you are seeing a lot of cells are there. Few cells are round because they have died. So all cells we cannot expect them to be alive. Some cells will die. Though whatever you are seeing round are actually dead cells. Then most of them are elongated, that they are the attached cells. So let us just get to a higher magnification just to see how these cells are. So when you go to higher magnification, usually you need to change the brightness. So this is 40x magnification, actually not 40x, it will be 400x because the eyepiece will have a 10x magnification. So you can see the cells more clearly, how they are very much elongated. So I can even go to a different place in the flask, everywhere it will be same. So different places will have different number of dead cells. So it's a very dense cell, lot of cells are here, you can see it, they are all elongated. Okay, so these are the cells. The next step is we will add trypsin to detach them from this attachment at the bottom of the flask. Then we will transfer it to an Eppendorf tube. So we will do that, and I will show you the Eppendorf tube. Okay. We will not actually show the trypsinization to you because we have already like parallelly doing it because we have multiple cultures. So we will I will just show you the Eppendorf tube which has the suspended cells. Okay. So we have trypsinized the cells in the media. We have added fresh media. Okay. You can see the cells at the bottom. Was this centrifuged? Okay, these are not centrifuged, there is a process. Yeah, you can see this. This is the cells after trypsinization. We have added fresh media, so for the cells to take nutrients. This is, that's why it is pink in color, fresh media is added. This is the drug that we will be testing with it. Right now we have used a very high concentration of drug, okay, because of confidentiality purposes I cannot reveal what drug is this, but this is very high concentrated drug. This is not a practical concentration, because for practical concentration we have to test it for at least a day, because where do you see somebody eating a drug and suddenly getting cured of cancer? So that doesn't happen. So there is that, because if you use a high concentration, it will have side effects in your body. But for showing it to you in this module, just to show that the drug can kill the cells and see how that causes a change in impedance, which is the modality with which we are doing drug screening, I have used a high concentration of drug. Okay. And these are cells. So the cells are in suspension in the media. You can do a process called microcentrifugation. Okay, where you keep this in the centrifuge, it will rotate and it will collect particles depending on this density. Okay, so still you can see the cells getting collected at the bottom here. Lot of cells are there. There is a white pellet at the bottom. There is a white pellet at the bottom, which is most of the cells getting collected there. The media is there. Okay, and this sample we will be using for our drug testing. Okay, cool. So how are we testing it? We are using microfabricated electrodes. Correct. What modality are we using? We are using impedance sensing modality. What does it mean? So we add the cells, we measure the impedance. What is the impedance of the cell suspension? Then we add the drugs, okay, and we wait for like 1 or 2 minutes because the drug concentration is very high. This drug will kill the cells, okay. Some cells will die, some cells will not die. When the cells die, their cell walls rupture, their intracellular contents enter the medium. They are more ionic, so the overall impedance of the cell suspension reduces. We again measure the impedance. If we see a change in impedance between the point where no drug was added and the point when the drug was added, then we can tell that the drug is working. Okay, this is the basic simple hypothesis of our testing methodology. Okay, next step we will show you the microfabricated sensor as well as the testing setup. How are we measuring the impedance of the drug testing? Okay, and then we will directly start measuring it, and we will see how things are playing out. It will be a very interesting exercise. So hold on. So we are coming to the last and most exciting part of this module. We have made the setup to do the drug testing. Okay. So first thing, the core of this is the microfabricated sensor. So let's look at it. So we have the sensor here, which have electrodes. I think you have already seen this sensor before, so I will not get into the full details of it. So we have the electrodes, and we have made 3D printed wells and attached them to the electrode structure. So we have each individual wells where we will put our samples. Okay. So what we will do now is, to one of these wells, I will select this well. Okay. I will add our cells. Okay. First, I will add our cells into it. Then we will measure the impedance. Okay. And then after that we will add the drug to the same well. What will happen then? The drug will kill the cells. Some cells will die, some cells will not. This will cause a change in impedance of the solution that is in the well. We will again do the impedance measurement, and we will show that what is the difference between both these impedance measurements. So the difference, if there is a difference, then that means that this method can be used for a rapid drug screening process. Okay, ideally a drug screening test will be carried out for a period of like 12 hours. To show this for you, I have made, as I told before, I have made a higher concentration of the drug. Okay, so for efficient, for correct proper experimentation, you need to have a statistically significant experimentation process. So what we do usually is we repeat whatever we are doing in at least 3 wells. It's called n equal to 3 technical repeats. Okay, we repeat in 3 wells, and we do measurements from all 3 wells, and all 3 have to be consistent. That is how you do experiments. Okay, now for you to show it to you, we will do it in one well and show you the basic hypothesis working. Okay, what is the hypothesis? Impedance value measured of a cell suspension changes when the drug is added. That is the hypothesis. Okay. To prove the hypothesis, we will use one well. To show that the experiment succeeds, we need to have statistically significant results. For that, we need to do technical and experimental repeats. What is the difference between technical repeat and experimental repeats or biological repeats? Technical repeat is you use one sample, but you load this, do the same experiment on 3 wells. Okay, that is technical repeat. What is biological repeat? You repeat the same experiment on different time points using different samples. Even when using different samples, you get relatively same results. Then it means that your experiment is statistically significant, and experimental repeats and technical repeats are consistent. Okay, that's how an experiment is designed and executed. Okay, this is generic information. So let's start off. Okay, so just for a brief thing. So this is a micromanipulator. We have already covered this. This is the impedance analyzer. Micromanipulator, impedance analyzer. The impedance analyzer's probes are connected to the micromanipulator. This is the tip box where we have micro tips to load the sample. This is the micro pipette to load the sample, 20 to 200 microliter micro pipette. This is the control unit to control the micromanipulator. Okay, it is switched on now. Then finally we have the software. This is the software where we will be seeing the impedance results. Okay, so this is our test setup. Okay, you need to be very aware that everything is in place, including a glass cylinder to put your tips, waste. Everything should be pakka when you start doing your experiment. Okay, we have our samples here on this sample stand. We have our cell suspension. We have our drug here. We have our drug. Okay, cool. So let's start the experiment. Okay, what I will do is I will choose one well here. Now we will start the experiment. So what we will do is we will load our sample in one of these wells. Okay, and then see how the measurements are coming out. So let me take the third well here, or let me take the 8th well here. So I will take the 8th well here on well, and we will load the sample. Okay, I will set the volume to 30 microliter. So we are changing the micro pipette to 30 microliter. Or let's keep it at 40 microliter. So we take the tip, pipette tip, we take the sample, open the sample. So before we do that, as we all know, the cells are at the bottom, you can see the pellet. So let me just shake it up a little bit so that cells are in the suspension. So now cells are properly in suspension. Before they settle down, let's take the sample. So the sample is there in the tip. Sample is there in the tip. Now I am going to load it. I will add another 10 microliter to it because there is still volume in the well. Let me add another 20 microliter to it. Yeah, now the sample is loaded and we are ready to take our first measurement. So let me just connect it. So I am focusing it on the contact pads. So he will. So we have my lab mate with me here, Arun. So he will just adjust the this micro manipulator so that it moves down. Please adjust it. So you have to make sure that you have a very strong contact with your contact pads. Otherwise, your measurements will not come out properly. Now this contact is made with the reference electrode. Now we have to go to the electrode of the well, which is well number 8. It has what happened is that it has reached the end of the set direction and still could not make contact. So these things will happen. What we have to do is we have used this as micro manipulator, right? So we have to push it down further a little bit so that we can reach the bottom. So we have adjusted the height of the contact so that it will reach the bottom. So now he is adjusting it again. Yeah, come down further. Please come down again. Keep coming down. Yeah. So we have made contact. So let's just confirm the contact of the reference electrode also. Yes, both are nicely in contact now. Okay, for this well, you can see the pink color where the sample is loaded. The electrodes are there underneath and the contact pads for measuring from this electrode are in place. We have connected the impedance analysis probes on those contact pads. So now it's time to measure. So let's look at the software. So now this is the interface as you might have seen in previous lectures. So for me to show you both the differences, so what we will do is we will measure both impedance and angle. So first item is impedance, it is selected. What we will do is we will use an excitation voltage of 10 millivolt and we will also measure angle. What is angle? The imaginary angle for the complex impedance with which you can resolve the impedance into resistive component and or capacitive and inductive component, which is the R plus j omega c or R plus j omega 1 by j omega L, 1 by j omega c or R plus j omega L. Okay. So then what we will do, we will start with 0 frequency and we will end it 100 kilohertz and we will take 200 points. Okay. So when we click draw, it will start acquiring the impedance data. Click draw. Yes, so it is taking the data. So we are having the measurements being carried out. So it will take time because it is spanning through frequencies. Because it is an impedance analyzer, it takes the impedance values from a range of frequencies which we specify. Here we have specified from 0 hertz to 100 kilohertz. So, the green color that you are seeing on the screen is the impedance value, and the red color is the phase information. You can see the real time impedance value plotted with green and the phase information plotted with red here. Okay. Things will be more interesting when we add the drug and see how the change is. That's what I was thinking, why you are holding it. The problem is that if I measure this, no, we will not be able to see any change, because that guy also will come on this range only. What I will tell that we have got the spectrum. Now we will measure it on 10 kilohertz only. Nalla difference കാണാൻ പറ്റും.
10 kilohertz is not.
It's recording, so I should be speaking.
We will take out the audio. Yeah, nice. I'm not coming in this way.
You can come, no problem.
No, I don't want.
So, it has finished acquiring the data from 0 hertz to 100 kilohertz. Okay. So, the thing is that if you see, it has almost flattened out after 10 kilohertz, correct, it is almost same value. So, there is no utility in taking data from all these frequencies. So, let us do one thing, we will change the frequency that we are acquiring, stop and reset, and we will take from let us say 8 kilohertz to 10 kilohertz only. Start frequency will be 8000 and end frequency will be 10,000, okay, and then everything else remains same. So, we will draw again. This decision we can make only after doing a first sweep of the frequencies and see that where it is changing. So, we saw that there is a sharp fall in the value, and then so we are going to look at a more interesting frequency range. So, now we are plotting the impedance and the phase information from 8 kilohertz to 10 kilohertz. It will acquire 200 samples and then we will see. So, this is all still signal acquisition. So, it takes time. What is happening is we are acquiring signal data from real world and then trying to show it on a software. So, you should understand the engineering challenges involved in making such a system. So, even this much time is a big achievement from instrumentation point of view. Tobacco testing? It is there?
I put everything in the testing.
Oh, you put everything in the So, we have finished the acquisition of signals. So, keep looking at the software. Now, I will add 20 microliter of drug to the same well. You have seen the drug. Now, we will add 20 microliter of drug, 20 microliter of the drug. Come down, further down, yeah, wait. So, I have added 20 microliter of drug to the well. We have to wait for at least 2 minutes for the drug testing to have an effect. We will wait for 2 minutes. So, we have added the drug, we have waited for around 2 minutes for the drug to take effect. Now, it is the eureka moment. Let us do the measurement and see if there is a difference. So, these are very micro level measurements. So, even if we get around 10 ohms or 5 to 10 ohm difference, which is repeatable, that itself is a good achievement and a marker of drug efficacy. So, we are doing the measurement now. Let us see how things happen. So, it is getting plotted. So, this is the second after adding drug. Okay. Measurement after adding drug. So, another thing to note is that I have added only 20 microliter of drug to this. So, let us see if there will be any difference in the values. If you see a clear demarcation between the green and the yellow lines, which is the impedance, then we know that there has been an effect, and also the red and the blue lines. Only when it completes we will know. So, there is a small update that came. So, it is taking the measurements from 8 to 10 kilo ohms. So, as you can see, there is a clear difference between the green and the yellow lines, and there is even more stark difference between the blue and the red lines. What you can see is that, as I had told you, that there is maximum change happening from 0 to 10 kilohertz, and after that it is constant, right? When we looked at the spectrum up to 100 kilohertz, now then we change the frequency with which we are seeing it. So, then if you see, when it starts off, the yellow line is below the green line, okay, and there is a crossover point, after which the yellow line is above the green line, and at that point the impedance also, the ankles are also, there is a clear difference, fixed constant difference in the angle measured between the blue and the red lines. So, this you can see a significant difference of the signals between when we have added the drugs and before adding drugs. So, this is how we can, this signal we can use as a measure of drug efficacy. Okay. So, I think you have got a very interesting experiment, you have seen that. So, we have seen a very interesting experiment, we have seen how signals are changing and which frequencies to choose, so many learnings from this experiment. But this was a very short experiment. Ideally, this should not be carried out in this short time, but for educational purposes we have tailored the experiment in that manner. But ideally, this drug testing should be carried out at least with microelectrodes also for a period of at least 12 hours for a realistic concentration of drug to be used against the cells. We have used a very high concentration, which is not a realistic scenario. Okay. But even then, conventional methods take 7 days and up to 2 weeks to give drug screening results, which we can use this method, we can get it in 1 to 2 days. So, these are the advantages of incorporating engineering methods to solve biological problems. I hope this example of drug screening will stoke further interest in you to explore this field, which is a very budding and exciting field. Thank you. See you in the next module.